Pleased to introduce our first speaker, Joe Barry. He's a professor at the Carnegie Institute up in Palo Alto at Stanford. And he is, we're really lucky to have him kick us off on the kind of the fundamentals of where the morning is going to go, and also for the workshop participants where the week is going to go. He's kind of the grandfather of global ecology, the kind of connecting what's happening at the plant scale, the leaf scale, to what's happening at the global scale. And so I think that's critically needed from the day he started working on it to today to tomorrow. And I think that we'll learn a lot about the thought process and the evolution of the science and the scaling through Joe's presentation and his involvement in the workshop. Thanks, Joe.
Yeah, thanks very much, Josh. It's great to be here and great to see all these friends in the audience. What I want to do is, as Josh said, try and set the some of the background for what we're going to be talking about, which is mostly two new tools: chlorophyll fluorescence and carbonyl sulfide. These really fit into approaches that have been around for a long time. One is remote sensing, and that's Jim Tucker on the left at about the age he was when all this started, and just before, in the late 1970s. The photograph or the picture aside from that, to the right, to the left of that, is a January-July images of NDVI on the planet. And that was in about 1986. Jim actually convinced NOAA to rearrange the spectral bands that they use in their weather satellites so that we could gain access to the vegetation dynamics. This is Inez Feng, who really pioneered the use of combination of weather models and tracers to analyze the influence of biology on the biosphere. This is in 1982, and what she did was to take information from weather models on winds, incorporate that into mixing algorithms to reconstruct the seasonal dynamics and the geographic distribution of CO2 in the atmosphere. Inez and Jim Tucker put these together so that you could visualize the correspondence between the changes in the vegetation dynamics and the CO2 dynamics, which were being produced by the network that Dave Keeling started. And another element of this really got started with Piers Sellers, taking biological information, the parameterizations that have been developed by plant physiologists, and putting them into the land surface models, which were used in climate simulation. This helped us in giving us a better understanding of how these biological processes work, but it also helped in providing information on how better information on how the climate system was working, and actually better weather forecasts. So these are essentially the tools that have been used since the mid-'80s to analyze the interaction between the biosphere, the atmosphere, the physical climate system. And we're going to continue to use those, but we have had the difficulty of really understanding, getting the measurements to drive these, to understand these systems, constrain them, and figure out how they're working. And what we're looking at now are new tools and new problems. The problem that I think we're focused on here now is that carbon dioxide is going up as a result of human activities. The carbon dioxide goes into the atmosphere, advances the greenhouse effect, and is a deleterious effect to the human activity. The interesting thing is that not all the carbon dioxide that we produce winds up in the atmosphere, and that could result, depending on how much it results, how much stays in the atmosphere, can result in different impacts for the same amount of human activity. These graphs show modeled trajectories giving the same CO2 forcing with different models. And what you see is that the CO2 concentration is increasing, but the increase is different for different models. This is really shown well here, where we're looking at the difference between the models. Some models predict CO2 concentration goes up fast, some slow, and this is due to different amount of sequestration, different operations of basically the carbon cycle, and this is a large societal driver of what we're thinking of. This is all really related to this really complicated carbon cycle, but if you think about it, that carbon cycle is really pretty much driven by photosynthesis. If we didn't have photosynthesis on the planet, most of these processes wouldn't be here. So photosynthesis is the primary driver of this, and in some ways, the most important part of it to understand if we're going to put all these pieces together and understand how they interact and work. We don't actually do photosynthesis very well, particularly when we're doing modeling in a one-if mode. That is, where we let the models run independently as opposed to using data to really constrain them. The dark line here is a model which uses data from the FluxNet system to constrain a data-driven model. The red are independently driven process models which are trying to reproduce the same thing. The actual data-driven estimates of GPP, this is the total GPP by latitude, and these are zonal sums. The process models catch the actual within their range, so we're not totally off, but the accuracy with which they reproduce photosynthesis in the global system is really rather poor. This is just independent models here summed to the globe. So not only do they miss in latitude bands or in spatial areas, but even add it all up, they differ from data-driven models. So chlorophyll fluorescence is a new remote sensing technology that's come about, but it's actually had a fair history. 1975, people recognized the measurements could be made of fluorescence in the presence of sunlight. The problem that we have when dealing with fluorescence is it's re-emitted light, and it's coming back to us from any sensor together with reflected sunlight. So distinguishing the two is a problem. Louis Gantier was able to accomplish this in 2007 using the MIRS satellite. And in 2010, 2011, Christian Frankenberg and Johanna Joiner's groups were able to get fluorescence out together from separated from the reflected light by using the GOSAT satellite, and we're able to show that the pattern of fluorescence matched the pattern of vegetation, and even more, that the total the radiance of chlorophyll-induced, solar-induced chlorophyll fluorescence matched GPP. This is GPP from that data-driven model that's produced by the Max Planck Institute at half-degree monthly intervals. And this is solar-induced fluorescence aggregated to half-degree intervals on monthly basis. And there's a very good correlation. It's about as good as the Max Planck does in reproducing flux sites. So it looks almost as if they had incorporated fluorescence into their model. It gives us something to work with. And I want to first try to explore some of the basis of that. Fluorescence of chlorophyll is a property of the chlorophyll molecule. Essentially, in photosynthesis, what happens is light is intercepted. This is a blow up of a unit which does the photosystem 2 component of photosynthesis. It has a central component that does the photochemistry and evolves oxygen. And that is driven by energy which is captured in proteins which surround this central core, which absorb light and move the light energy as an excited chlorophyll state moves from chlorophyll molecule to chlorophyll molecule, ultimately hitting the reaction center where the photochemistry can occur. Any place along this line, this pathway or random walk that the energy takes to get to its endpoint, there's a certain probability, kind of like the radioactive decay, that a chlorophyll molecule will essentially collapse to the ground state, re-emitting a photon. And that's where the solar-induced fluorescence comes from from plants, and all of the chlorophyll that's on the surface of the land is really associated with leaves. So first, the chlorophyll fluorescence is actually quite closely linked then to photosynthesis. We're essentially looking at the life of an excited state in the chlorophyll that's absorbing light. And the fluorescence tells us something about not only how much light has been absorbed, but the lifetime that a excited state takes to get from where it's absorbed to where it's used. And it's used in a reaction center, or it can actually be consumed by thermal reactions in other locations. So it's not only is a very specific remote sensing tool, but it also is gives us information about the process that's going on in photosynthesis. Just quickly, this is the spectrum of fluorescence plotted together with the absorption spectrum of chlorophyll. You can see that it's shifted to the red. It's normally measured here in an area where there's relatively small absorption due to chlorophyll, but it also overlaps with the chlorophyll absorption. So you might expect, and we'll see and talk in Christian's talk, how this overlap influences the spectrum of the light that's emitted and how that's related to the amount of chlorophyll in the system. The key to getting the chlorophyll fluorescence out of a satellite retrieval or actually from ground measurements relies on the fact that fluorescent light is new. It doesn't it's smooth, it comes out in a very smooth spectrum, whereas solar light is old. It's coming through the solar atmosphere and through the Earth's atmosphere, and there are a lot of absorbing compounds in those media which cause the solar spectrum to be rough, the fluorescence spectrum to be smooth, and using statistical techniques and spectroscopy where we can resolve the features, the rough features of the emitted light, you can deconvolve it into the smooth and the rough components. I'll not go into the details of that. Christian will do that. But the basic idea is that we can use this separation of light being smooth and new or rough and old to separate all the reflected light will have the roughness in it. All the fluorescent light will be smooth. So just to summarize here, it's really specific to vegetation. It's a bit different than reflectance. This is a reflectance image and this is a fluorescence image. You can see that in places where there's no green, it's black. And that means that areas that don't have vegetation aren't contributing to the fluorescence retrieval, but they do contribute to the reflectance retrieval, which has been our major driver, or the major workhorse of doing biosphere studies. So reflectance sees the whole scene. Fluorescence is really specific to the vegetation. It's also less sensitive to atmospheric scattering effects, which change the spectrum. And of course, the radiance of solar-induced fluorescence was showed in that other slide, and was showed almost immediately that when it was nicely detected, correlate with GPP. Just to put us into perspective or into what's kind of the end message with respect to fluorescence is that there are really two parts of this. The part of it that we focused a lot on is physiology. That is, these are model simulations, and these two lines correspond to imposing different fluorescence yields to the model. This axis corresponds to imposing different leaf angle distributions, different leaf area indices on to the canopy. So there's a portion of the solar-induced fluorescence that comes out of the system, which is due to physiology, causing the coupling between absorbed light and emitted light to change. And there's a portion that's due to structure, which has to do with the exposure of leaves, the capacity of leaves, and other things that affect essentially the probability of fluorescence once it's produced of getting back out to be detected.
So that's detected, that x-axis, by near-infrared reflectance? Is that what that is?
This would be by the near-infrared, at the 740 peak. You would also see, if you looked at the 680 peak, some effect due to chlorophyll content as well. So just to back up and think about the physiology part of this now, this is an antenna complex. It has a bunch of chlorophylls in it, which are nicely positioned so that their absorption vectors overlap, and so that they can, once absorbed, light is delocalized among many absorbing molecules. This system donates excitation to a reaction center, which is composed of two chlorophylls very closely positioned next to each other, which undergo a charge separation, and the electron is withdrawn from oxygen on one side and donated to a plastoquinone on the other side, and this initiates the first step in the flow of electrons in photosynthesis. Now, this process is fairly high octane chemistry. Very highly oxidized compounds are formed in this. If they're not quenched rapidly, photodamage can occur. through formation of superoxide radicals and other things like that, and that this system can be damaged. So you don't want to have this system sort of backed up. That is, when there's, say, more energy coming in than can be disposed of. So what biology has done is to evolve a system which allows feedback to occur from systems downstream of this whenever there is a backup. This system goes into a mode where excited energy is actually converted to heat within the antenna pigments. And this is called non-photochemical quenching. It behaves a lot like, we're probably all familiar with stomata and how stomata regulate the loss of water that is a cost of doing photosynthesis. In this case, these are stomata for light, and they regulate the loss, the cost that would be associated with having too much energy flowing into the reaction center complex in the core. Just to visualize this a little more, we have the classic discrimination of light reactions and dark reactions in photosynthesis. These are driven on this side by light, this by CO2 and temperature, and the amount of enzyme that's available. And we think of this as kind of like a teeter-totter. It flips from one limiting state to another depending upon the balance of the capacities of these two. And years ago, we made a model that just said you could model this by having a model for the light reactions and a model for the dark reactions, and you take the minimum. But that, while it solves the rate equation, it doesn't tell us how that happens. What did we do? We skipped over. I skipped over some. I must have put the wrong version of things up here, but I skipped over a couple. But let me go back here. We have capacity using PAM fluorimetry to drive this to model or measure what's going on in the light reactions, and we have enzyme parameterizations that we can use to evaluate the capacity of these steps, and we can figure out, using those two approaches, which one is limiting under any particular condition and how things shift from one area to another. And we can see that as this teeter-totter flips, this system goes from providing most of its energy to the reaction centers to providing less and less as the feedback becomes stronger and stronger. And we can see that if we change the capacities of reactions here by modifying the CO2 concentration or the amount of the CO2 fixing enzyme, we can regulate, or the plant regulates the amount that can be donated by this system. So those two interact. And that's what this shows. This is just photosynthesis versus light intensity for a plant that has low amount of rubisco and more and more and more rubisco. You can see that this is the photochemical yield. Photochemical yield is always dropping as light goes up. These are model results, but this is what you'd see with measurements. And it drops less severely with plants that have a lot of rubisco than with plants that have a small amount of rubisco. What we actually find is that during this phase where photosynthesis is going up, fluorescence also goes up, But then once it reaches this point where photosynthesis can't increase anymore because of carbon metabolism limitations, then the system starts to back up and this non-photochemical quenching cuts in, and the yield of fluorescence, which had been increasing, now goes down. And this pattern is reproduced, but over a wider range of light as the photosynthesis, as the light intensity is changed with different amounts of carboxylase. So one thing that we see is that fluorescence yield is linearly related to what we see in the rate of photosynthesis over these ranges where light is essentially limiting. And then it becomes nonlinear and starts to fall when you get into these areas where photosynthesis is constant with light, but of course the input of light energy is changing directly with light. This has, when this is translated into the fluorescence yield times the incident par, you get somewhat linear relationships, particularly in this region of low intensity, of limiting intensity. But once you get back out, get into this, it falls off to still being proportional to light, but at a lower slope. So you have a region here where it's nice, and a region where these diverge. It turns out that most of the remote sensing that we do is sort of in this range of the light responses of leaves. So that explains why we're seeing this nice correlation. We do see that when stress occurs, that fluorescence declines, and that's because we're going from, say, a curve like this to a curve like that, because stress causes, in effect, the amount of rubisco available to fall. So that gives us the sort of a background of the interaction of fluorescence with the physiology of leaves. So we've linked fluorescence to the conventional models of photosynthesis by using the PAM fluorimetry measurements, which Christian will discuss in more detail. It really requires one more additional parameter to be put in those models in addition to the dynamics of fluorescence generation. And that is a parameter which controls the response of the non-photochemical quenching feedback on the light reactions. So this is plants can actually live without non-photochemical quenching. You can put in inhibitors which block it, but it is a protective mechanism which prevents this damaging photoinhibition to occur. The amount or the extent to which plants use non-photochemical quenching costs them some in energy efficiency of doing photosynthesis, but they gain in the efficiency, in the resilience of the photosynthetic mechanism. So that's where we are with that one. Rubisco affects the amount that we see, and we've put this into several models, SCOPE, SIB, CLM, so that this process does live in the models that link back to, say, Seller's SIB. I'd also wanted to just mention, go back to this, that we have the NIRV. We have this structure component and this physiological component. We've talked about the physiological component. We're still working on the structural component. The interesting thing is that it seems to be very closely linked to the near-infrared reflectance of the canopy. And if you think about it, a solar-induced fluorescence photon is a near-infrared photon, and the probability of it escaping the canopy is very similar to reflected near-infrared photon from getting out of the canopy. So the two differ, or the two are quite the same, but canopies differ a lot in the near-infrared reflectance that they generate. And that isn't just a function of their fractional absorption of PAR. You can see have a canopy, for example, from a conifer which is very dark in the near-infrared, or a canopy of, say, sunflowers, which are very bright in the near-infrared, absorb the same fraction of solar incident radiation. And they, interestingly, have very different photosynthetic rates and very different near-infrared reflectances. If we put just SIF or this near-infrared reflectance into SIB, we can, as a replacement, making the integration factor proportional to these inputs, we get much better simulations of the seasonal cycle and the quantity of fluorescence. This is at Harvard Forest. These are improvements we see at other sites. So there are ways in the future, I think, that we'll be able to use fluorescence to actually drive the models as well as to test the models. So it's turning out to be surprisingly useful. Indicates beginning and end of growing season, drought. It's also really been an important area in photosynthesis community, so it gives us a way to tie into another scientific tradition. This NRV is a good proxy for SIF, and it would probably be better to use SIF if we had an appropriate satellite, but we really lack the kind of satellite that we need. And it's going to link to COS exchange, which we think also looks at photosynthesis. This is just my attempt to solve the satellite problem. This would be a geostationary satellite located over the Amazon area. So we got to just maybe going to shift gears right now if there's really a quick question that somebody has. Otherwise, we'll flip to COS. Okay, COS is something that we can use to augment our measurements of CO2 concentration from the atmosphere. It's carbonyl sulfide, which essentially has a CO2, is similar to CO2 in structure, but has a sulfur in place of one of the oxygens. The red lines here are CO2 changes in the atmosphere, and the blue lines are changes in COS concentration in a vertical profile and at Niwot Ridge year by year. So the seasonal variation is really quite large relative to the background concentration. This just shows the same thing. The same pattern exists, but if you look at the concentrations, the relative change in COS is much larger than the change in CO2. This just plots one against the other, and the ratio relative to background is about 6 to 1, between 6 and 10 to 1. Larger signal in COS due to the biological driver of the biological driver on atmospheric CO2 concentration. So COS, even though it's present in low concentration, seems to have a larger relative signal than CO2. Just a couple of quick things on the properties. 400 ppm, 500 ppt, so that's 1 in a million CO2s one COS per million CO2s. It's a low concentration, but it's easy to, it can be measured. Turnover time in the atmosphere, it's mixing, is very similar. It doesn't have a large flywheel. It's not linked to an ocean reservoir like CO2 is, so that it actually changes when the fluxes change on a 2-year time frame. CO2 can change. Rapid changes are similar to those in COS, but then it's linked to the exchange with a large reservoir in the ocean. It has we have a fairly weak signal on uptake, and that's because CO2 is always mixed together with respiration. So you have CO2 production and CO2 consumption locally. Uptake and production seem to be geographically very separate for COS, so that you see a much stronger signal, and that's why you see that. It involves rubisco for CO2 and carbonic anhydrase, which is another enzyme involved in CO2 metabolism in chloroplasts, does the COS uptake. This is basically the idea. This is a leaf. These are the resistors that are involved in movement of gases, ultimately to where they're consumed by biochemistry, from where they start out here in the atmosphere. We often refer to this as COS, but often also to OCS, and you'll see me using both here. The pathways are identical. There are drawdown in concentration along the way, and that's why this is a good proxy for photosynthesis. It's essentially following the same route from out in the atmosphere. This is just the experimental system that Karen Stimler and Dan Yakir, some almost 10 years ago now, started at the Weizmann Institute looking at the gas exchange of COS by using a QCl. It is generally follows photosynthesis, but it actually deviates down here at very low light intensities. This is the ratio of them. And that's because COS actually doesn't have any energy input requirement. It's just following the response of stomata to photosynthesis. And that is determined by, in many models, by just the CO2 concentration and the water vapor concentration and a proportionality constant to the rate of photosynthesis. But there is a residual stomatal conductance that doesn't go away at zero light or at zero photosynthesis, and that's what causes this divergence at low light. Very quickly, just we can code this by comparing the conductance of CO2 to that of water for the stomata, for the boundary layer, and for the stomata to water. And then we have a conductance due to the cell, which turns out to be quite linear if we plot the uptake of COS to the intracellular concentration of COS. So we can come up with an expression which essentially uses assumes a relationship between the intracellular uptake and a mesophyll resistance, which includes both a physical diffusion and a biochemical component. These are just light responses taken from Stimler's work, temperature responses. They work pretty well. They can be incorporated in larger scale models, and this is just a diurnal patterns of CO2 and COS exchange, showing that they're similar, except that CO2 is being produced at night, taken up during the day. COS is being taken up all the time because there is COS uptake by the leaves and also by the soil in these canopies at night. You can think of these as being driven by processes. You can think of the atmosphere as being pulled around by, say, photosynthesis taking out COS and CO2. This is CO2 increasing in this direction, COS increasing in this direction. Photosynthesis pulls it this way. Respiration would pull it this way. There's some soil production, it would push it that way. the net tells us something about how these processes are working in combination to affect a local or a regional change in COS concentration together with the CO2 concentration. If we think of this at a global scale, we've got production from the ocean, we've got the anthropogenic fluxes, respiration, some photochemistry going on in the atmosphere, photosynthesis, and these integrate, or the vector sum of all of these should be come to zero if the state concentration is stable over time. Just quickly, this is a global model which we've put together. Biggest thing that we had to do to make this work was to put in an ocean source and to use the biology, what we think we know about photosynthesis to drive the COS uptake, and we can get it to match atmospheric concentration measurements at various sites around the world. And I wanted to just end with this, which is a simulation using those surface fluxes. This is a transect along the equator in January. Blue is low COS concentrations, red is high COS concentrations. And what you're seeing is that deep convection is moving material from the surface, either COS that's coming off the oceans or COS uptake that's occurring over the continents and pushing it up to the top of the atmosphere. And I think we'll see later that this is related to, we can relate this to what we see with satellite measurements, and hopefully gain some insight both into the processes that are occurring underground, but also to the atmospheric processes which are mixing COS in the system. So I'll end with that, and maybe there's a bit of conclusion there, but maybe we could start with questions while you just read those.